Okay, so we start 1.2. The title is Ultrastructure of Cells. Now, first of all, when we talk about cells, you know that they're really, really small. So how are we able to look at cells? We have a lab. We had a lab, and we actually made use of a light microscope, and we were able to somewhat see the structure of the cell. However, if you use a light microscope, it cannot really reveal what's inside, what are the organelles present. Now the title is Ultrastructure of Cells. Ultra, which means super, extremely, or a very, the very structure of cells. So if you just want to know the overall or the general structure, you can use a light microscope. However, to reveal the ultra, the detailed, the extremely detailed structure of your cells, we should use an electron microscope. Now, the reason is the light microscope is limited in its resolution. So no matter how bent your lens will be, it will always create a blurry picture. It's because it's explained in the book. If you don't understand, it's okay. The wavelength of light is between 400 to 700 nanometers. But what you're looking at is smaller than that range. So how can you use, how can light reveal the image if it's bigger than what it's measuring? However, electrons are very small. So if you have this image and you shoot it with electrons, you keep shooting it with electrons, you can get detailed images of all of these little parts of what you're looking at. So electron microscope can give you a higher resolution. Therefore, an electron microscope will be able to reveal the detailed structure of your cell. Now, your cells can either be prokaryotic or eukaryotic cell. Eukaryotic. Pro means before, karyote means nucleus. So these are cells that doesn't have a nucleus. These are structures that don't contain a nucleus. However, eukaryotic cells, eu from the word true, karyote, nucleus, these are cells that contain nucleuses. So how do we describe the structure of a prokaryotic cell? So a structure of a prokaryotic cell, number one, they're simple in structure. They do have a membrane. In fact, some prokaryotes also contain a cell wall. They also have a ribosome. However, it's smaller, so the size is about 70 Svedberg units. They do not have a nucleus, but they have a genetic material arranged in an area called the nucleoid. Okay, and if I ask you to either label a prokaryotic cell, or if I ask you to draw a prokaryotic cell, I'm going to look for these things. So let's look at an example of a prokaryote. We'll look at it an E. So as you can see here, it has a plasma membrane, this purple one, and then an outermost covering the cell wall, the red one. So cell wall and plasma membrane. Then it will have its genetic material free floating in the cytoplasm. There's no nucleus. So this area is called the nucleoid. And those pink dots or circles are your ribosomes. It will also have hair-like projections called your pili, and this longer projections called the flagella. So as you can imagine, the flagella is for swimming through the medium or for movement in water, and then the pili are for attachment, to attach to a tissue or to attach to another bacteria. So this is the general simple structure of your prokaryote. Again, it has cell wall, cell membrane, the nucleoid region, your ribosomes, the long flagella, and these little hair-like projections called your pili. Bacteria are very simple. So in fact, even their reproductive method is also very simple. So let me show you here. I'm going to use McGraw-Hill animation. So how do prokaryotes reproduce? So they don't need sexual gametes. What happens is the cell elongates, the DNA replicates, and then it will pinch off in the middle. There you go, dividing it into two. So that's it. That's how they reproduce. It's a very simple process, and we call this binary fission. Fission from the word to split. Binary means two, to split into two. Okay, now for your eukaryotic cells, these are more complex cells, and your eukaryotic cell structure shows compartmentalization. So what is compartmentalization? What are compartments? Compartmentalization. Compartmentalization. So to compartmentalize means to have different, distinct, and separate compartments or sub-compartments, sub-parts. And you know that your eukaryotic cell are made up of organelles, each with their own membrane. So organelles ranging from your nucleus to your RER to your smooth ER, to your Golgi, to your cytoskeleton, etc., etc., etc. What you need to review here are the function of each of those organelles and also how it looks like under the electron microscope. I might give you sample pictures, sample electron photographs of these organelles, or I'm gonna ask you to identify these organelles. So have to remember that the organelles present in your cell is dependent of the function of that cell. So for example, there's two examples used by your textbook. So we're looking at a cell here from the pancreas, and you already know that the pancreas, it's an organ right beside your stomach. It's in charge of producing different types of digestive enzymes. So enzymes are proteins. So if it needs to produce lots of proteins, lots of digestive enzymes, it means it needs to make proteins. So these proteins, these enzymes, after it's made by the ribosomes and modified into Golgi, it will be packaged inside these tiny red dots. These are vesicles or secretory granules, secretory vesicles. Another example used by the book are your Palisade mesophyll cells. So as you've learned, the Palisade mesophyll layer is the layer where you have cells that are in charge of photosynthesis. So if you look at one Palisade cell, what you can see is there's many right here chloroplasts. And why is that so? Because the chloroplast contains chlorophyll pigment that can absorb light. Therefore, this cell is actively involved in photosynthesis. Another example that is worth mentioning are your muscle cells. Your muscle cells will obviously contain large amounts of mitochondria since it needs a large amount of ATP to contract. So the point here again is the presence of organelles in the cell is dependent on the function or the use or the role of that cell. Okay? So as you study this chapter, make sure that you familiarize yourself with how the organelle looks like under the electron microscope. So your Golgi here, it looks like flaps of membranes. Your lysosomes are the tiny circular dots. Mitochondria it's spherical with membranes inside. Okay, and then then you have your chloroplast. It's spherical as well with stacks of a granum. And then let's see. That's it. Here's another example from the book. From here, so you have your spherical organelle, that's your mitochondria. The bigger one is your nucleus. It's pointing at somewhere and you don't exactly know what it is. Your good guess would be a ribosome or a cytoplasm. So this one is pointing at a free ribosome in the cytoplasm. And let's see. How do you differentiate a mitochondria from a lysosome? It kind of looks the same. So just remember your lysosome is darker stained because it has digestive enzymes. What else here? So we have this random flap of membrane. So it's your Golgi. And you have here another flap of membranes. It's not as clear if it's RER really. But you make the judgment call. So remember, what you need to study here are the different functions of your organelles at the same time, how these organelles would look like under the electron microscope.